Okay, so I'm going to kick off today's proceedings with a summary of yesterday's presentations and the discussion sessions. Okay, so yesterday's session started off with some introductory talks by Anna, Ilan, and Eric, and these talks just focused on providing a background to the adaptive and innate immune response biology. And then they discussed some FDA guidance. Obviously, the ANDA peptide guidance was discussed and introduced, and then they looked at some product-specific guidance for peptide products, of which we now have, I think, about 70 different ESGs for peptide products. The second session was on adaptive immunogenicity risk mitigation. This was chaired by Daniela, with speakers Narasimha, Annie de Groot, Rob Siegel, Sophie Tordo, and Mohan from the FDA. The discussion here focused on the challenges that are associated with adaptive immune response assay development. And in particular, a lot of discussion was centered around the validation of the in silico MHC, HLA binding, and T-cell functional assays. Specifically for the in silico modeling, this was discussed in the context of HLA binding to the peptide alleles, and then the T-cell receptor binding to the peptide epitope that's bound to the HLA. The challenge of demonstrating in silico, in vitro, in vivo correlation was also discussed, and then the limitations in terms of modeling of the peptide containing non-natural amino acids or covalently attached side chains was also discussed. And this is an ongoing area of research. The discussion continued around the harmonization of protocols for MHC binding assays, also the MHC-associated peptide proteomics or MAPS assays, and of course the T-cell proliferation assays. In terms of challenges, some of these were identified as the synthesis of test peptides in appropriate purity, the choice of cells in terms of the population, the viability of the cells, the functionality, the number of cells per well, and for example, PBMCs was obviously a cell line that's used, or a cell that's used extensively. In terms of the number of donors, the diversity of the donors, whether it's healthy versus patient, these were identified as factors that are important to ensure a broad HLA coverage. Selection of controls, factors that were important here were the length of the peptide, the purity, the propensity for aggregation of that peptide, T-cell epitope content, and endotoxin content. In terms of defining the assay sensitivity, selectivity, and positive signal acceptance criteria, this was another area of focus. And then there was extensive discussion around the appropriate concentration for the peptides in these assays. And there was also some discussion about the acceptability of the current immunogenicity related impurity thresholds that are recommended in FDA guidance as well. And then the second session moved on to a discussion of innate immunogenicity risk mitigation, and this was chaired by Eric, with speakers Andrew, Jeremy, Sophie, Noel, Seth, and Daniela. The discussion here focused on those challenges that are associated with development of innate immune response assays. In terms of the types of assays that were discussed, there was some discussion about cell line assays, the choice of cell line and the readout. So for example, HEC blue, raw blue, THB blue cell lines, and NF kappa B is a potential readout that can be used. But most of the discussion focused on the cytokine assays, as these seem to be more favorable these days for conducting these types of assays. So these assays can be conducted using primary PBMC cells, or whole blood, and the choice of fresh or frozen whole blood was also discussed. The advantage of these assays is that you can measure multiple cytokines in one cell. In terms of challenges, the number of donors was also an issue here, as it was for the adaptive immune response assays. Usually healthy donors are chosen, and it's based on inter-donor variability, the number of donors. Cell viability is also important, especially in the context of the formulation. That's important to the assay sensitivity. So there was a lot of discussion around preservatives such as phenol or metacresol that are often present in these types of products and how that can interfere in the viability of the cells. So things that are important to consider the appropriate dilution to minimize this effect to a concentration where those interfering excipients are not causing cell death, essentially. And so as a way around this, the concept of testing the drug substance to avoid this effect was also discussed. In terms of validation, it was discussed that it's important to validate sensitivity of the assay to the IMIs that may be present. The choice of positive, negative, and suitability controls that are critical to demonstrate assay sensitivity, and also to ensure a broad coverage of the innate immune response receptors. Acceptance criteria was discussed, and justification for choice of the statistical approach for data analysis in these assays was also extensively discussed. And there were some key questions. Are the detectable levels of ERMEs clinically relevant? Are the cell lines sufficient to discern ERMEs, and why multiple biomarkers, advantages and disadvantages, all these were discussed. But then in the afternoon, late afternoon, we moved on to the in-person discussion sessions, and I'll just summarize the key themes that were discussed. So for the first session, we focused on adaptive immunogenicity risk mitigation, and there were questions around the differences in results generated using different in silico models and how to validate these types of models. The DCT assays were discussed in terms of things like culture time, control of cell viability, number of cells or CD4 plus T cells, and ratio of DCT cells in the well, establishing assay sensitivity, FPS or defined media, cytokines and growth factors. Again, there was a session discussing the appropriate concentration to test individual impurities. That's been quite a hot topic. Improvements to current assays that could be made to better reflect clinical immunogenicity, so trying to make that clinical bridge in a more robust way. And then another discussion session on donor selection, so determining the number of donors by HLA coverage, assay variability or both, and whether to use healthy donors or patient primary cells. And then in session 3B, this one focused on the innate immune response theme. First discussion session was on critical assay attributes for establishment of the assays, and whether more sensitive or relevant biological readouts for innate immune activation. The main challenges in implementing these assays to assess the immunogenicity risk for generic peptides and helpful strategies. So this was more of a general catch-all for these types of the problems that are observed in these types of assays or developing these types of assays. There was another session discussing the strategies to overcome excessive dilutions due to formulation inhibitory effects, which I mentioned earlier. And then there was a session discussing the statistical analysis approaches to analyzing this type of data. There was also a question around the criteria to accept or select assay runs and ERME data and qualifying attributes for donor selection. And then the last question was was a sort of a hypothetical situation of how to proceed if drug product batches show a signal in one or more of the ERME assay readouts compared to the RLD. and the need to identify MEs in order to implement controls. So I'll just say that we've collected all of the notes from these sessions in the afternoon, and we will be compiling those into some proceedings for the meeting that will be published in some form after the meeting. So yes, I will finish up now and hand it over to the next speaker, Steve Kozlowski. I believe will be joining us virtually. Thank you.
Good morning. Is everybody able to see my screen? All right. I want to thank Tim for that very interesting summary of yesterday. Obviously, a lot of important things discussed, and I want to give an introduction to day two, and I want to thank the organizers for inviting me to do that. So I will start by going over something that may have been covered a little bit yesterday, but a little bit about the history of MHC. Sort of, to me, a scientific puzzle over decades that overlapped with my entry into the world of science. That the idea of genetic control over immune responses was always something that there was great interest. And in 1970, there was this discovery of the association between histocompatibility, antigen markers, and immune responses. And initially, that was really felt to be a linkage, and there was probably some hidden T-cell receptor gene in that locus, and not necessarily that histocompatibility and immune responses were directly linked. Over time, over the next few years in the 70s, the idea that there was something called MHC restriction, where the target cell had to match an MHC for T cells to be able to operate, began to make the idea a little more complicated. And later, immunoprecipitation, more correlation really began to say that this genetic linkage is identity. And at that time, and I was actually a summer student at the NIH in 1981, there was a great race to who could show that antigenic peptides actually directly bound MHC. Fast forward, in 1985, it was shown, right, that you could directly bind peptide to MHC molecules. Shortly after that, the crystal structure that really showed the picture of this linkage. And then shortly after, beginning to elude all the different types of peptides, in particular MHCs, and understanding the pockets and which part relate to MHC and which part relate to T-cell receptors. Better understanding, therefore, the role of the T-cell receptor. Still, antigen processing was important, and ultimately, this concept of how T-cell adaptive immunity really works. And last but not least, as a big discussion yesterday, the idea of danger signals and innate immunity and how that impacts all these things. So this is science with a 50-year history, and it's very, very exciting that this is beginning to have more and more of a practical role in how we make decisions. So if one is concerned about the immunogenicity of a product, one has to be able to link the actual product that the manufacturing process generates and understand its efficacy, safety, and immunogenicity. And historically, clinical trials were the ideal gold standard and probably only way of really being able to make that. And in certain circumstances, immunogenicity is really the only uncertainty. And so clinical trials just for immunogenicity are a very expensive and inefficient way to develop products. And so figuring out how to take adaptive immunity and innate immunity and make up for this so the clinical trials are not necessary is really a tremendously important goal. And many tools, whether in silico, in vitro, and non-clinical, are available, and many of those were discussed yesterday. Today's session and sessions really are going to be focused on extending that into two particular areas. Recombinant peptide products as opposed to synthetic ones, and oligonucleotides, a really growing product area. And the session will really start with looking at host cell proteins and their roles in that. So Monsi Puig will be chairing that session and looking at the regulatory pathways that can manage recombinantly manufactured peptide products and the strategies for mitigating that risk, which is often associated with host cell proteins that may not be present in synthetic peptides. So Sophie Shubow will give the first talk about how in vitro data can be used in immunogenicity risk mitigation for recombinant peptides. Vibha Jawa will then follow with a risk assessment of host cell proteins related to these products. And Emilee Knowlton will talk about MHC-associated peptide proteomics for characterizing peptide epitopes. After that, there will be a coffee break for you to think about and consider all the things you've learned about and be energized to come back, where around 10:25, alternative models for predicting immunogenicity will be discussed in sessions. Kristina Howard will talk about a broad range from humanized mice to microphysiological systems to predict immunogenicity. And we'll hear from Nico Gaudenzio, I hope I'm pronouncing that right, about a new in vitro framework for assessing immunogenicity using injectable human skin modules. After that, there will be a Q&A session with the panel and then lunch from 11:35 to 12:40 p.m. We'll then move to session 5, which will go from 12:40 to 1:50 p.m. Dr. Di Zhang will chair that session. And the oligonucleotide sessions will focus on the challenges of characterizing oligonucleotides and relating the sequence-related impurities to immunogenicity risk. And similar to peptides, the role of innate immune modulating impurities in these products. Markham Luke will give an introduction, and then Hu Yang will talk about impurity characterization in synthetic oligonucleotides. Then Hope Bart Rogers will talk about regulatory oversight of oligonucleotide therapeutics. And Sudhar Agarwal will talk about oligonucleotide structure and innate immune inactivation, as certain sequences of nucleic acids can themselves be direct innate immune activators. The session will continue, and again, focusing on characterization and innate impurities, immune modulating impurities. So there'll be a talk by Hana Lee about assessment of IMRI for oligonucleotides. There'll be a talk by Philippe Kolenk about safety considerations, including immunogenicity when developing generic oligos. And then there will be a Q&A session on that. At that point, virtual attendees we'll hear closing remarks. Actually, everyone will, but it will be closing remarks for the virtual portion by Rob Lionberger. And then, as there were yesterday, there will be closed sessions. Short coffee break before them till 3:35 p.m. And then these working group sessions will focus on host cell proteins and alternative models, sort of the morning portion of today's seminars, and on synthetic oligonucleotide therapeutics, what you'll hear in the afternoon. There will be closing remarks by James Polly and Anna Schwendeman at the end of the day. And so hopefully from today, there will be insights and learning into how to manage host cell protein impurities for recombinant peptides. There will be good discussion of alternative methods to predict immunogenicity and complement the tools that have already been discussed. And lastly, there will be good discussion on oligonucleotides, a growing form of therapeutics, and one which has immunogenicity considerations and really how to move forward with what's been learned from proteins and peptides in this area. And so with that, I will turn this over to the next speaker or moderator. Thank you very much.
Good morning, everyone. I'm very excited to get the first session of today started. Today, we're going to be talking in this session about host cell proteins as a new category of impurities that they were not discussed yesterday because they pertain to recombinant peptide products, and also alternative models to assess immunogenicity. So although peptide products recombinant peptide products can rely on the findings of the RLD of safety and efficacy, the fact that recombinant systems are being used, cellular systems are being used to produce these peptides, that can lead to differences in the product quality attributes of the generic products compared to the RLD. So it's very important to take this into consideration because impurities such as host cell proteins derived from these in vitro systems can be impacting immunogenicity as well. So part of the session is going to be dedicated to discuss how to assess and how to mitigate these host cell protein impurities and the effect on immunogenicity. Also, we heard yesterday that we have different systems in silico, in vitro strategies to assess immunogenicity. But we also heard this morning from Cameron when he made the summary that we have challenges on how to interpret this data and how we use this data moving forward on the speculation on how these impurities can affect the immunogenicity clinically and also the relevant changes in its safety and efficacy. So today we are going to hear on the second part of this session about alternative methods that we have, perhaps more in vivo and also using human skin modules that can bring the assessment to another level to make it kind of more biologically relevant. So, having said that, I'd like to invite the first speaker of this session. This is Dr. Sophie Shubow. She's a senior biologist in the Office of Clinical Pharmacology in CDER, FDA. She leads center-level immunogenicity review committees in which multidisciplinary teams are being involved in discussing approaches to immunogenicity risk assessment. She's also actively involved in groups that develop guidances and policy. So Sophie today is gonna talk about the follow-on recombinant peptide, role of in vitro data in immunogenicity risk mitigation. Those are conversations that are ongoing in the agency, and we hope it's going to bring some clarity to the standpoints. Sophie?
Thank you, Montse. I don't know if I will bring any clarity, but I will certainly ask questions. My brief presentation essentially aims at setting the stage for the future-looking discussions we will have later on how to leverage novel in vitro and analytical methods to better evaluate and mitigate the risk associated with these host cell proteins in recombinant follow-on peptides. This is my disclaimer. I will say a few words to start with about the processes that can be used to manufacture peptide products. I will then introduce a potential role that in vitro and in silico immunogenicity assessment, which I will call IVISIA, can play in the evaluation of follow-on peptides made using recombinant processes. And then I will shamelessly promote a Federal Register notice that FDA published back in July in order to collect more information from the public, including you, about how you control and evaluate the risk of these ocell proteins in your drug product. Increasing demand for certain peptide products, such as GLP-1 drugs, is likely to translate into more regulatory submissions for recombinant follow-on peptide products. On this graph, you can see that sales of GLP-1 products are predicted to almost triple within the next 6 years. While solid-phase peptide synthesis is well suited to the manufacturing of these complex peptides, it also generates large quantities of organic waste when used at large scale. And large-scale manufacturing would be necessary to meet the growing demand for these products. On the other hand, recombinant technologies are, for one thing, much more cost-effective, and for another, much more environment-friendly than solid-phase peptide synthesis. And actually, some of the manufacturers of these products already use these processes. The immunogenicity risk of follow-on, The immunogenicity risk of a follow-on peptide product is evaluated in comparison with that of a reference-listed drug or listed drug, as you all know. And most of these comparative immunogenicity risk evaluation is focused on product and process-related impurities. The main differences between a follow-on recombinant product and the product it references are in a subset of process-related impurities that are called generically ocell remnants and ocell proteins, which are essentially cell-based biological materials that from the expression system that will co-elute with the final drug product. And these residual ocell proteins are considered a critical quality attribute in the manufacturing of biotherapeutics, including peptide products. Why is that? Because we know that they can critically affect product efficacy and safety in multiple ways. In terms of product efficacy, so some ocell proteins are proteases, such as catepsins, that have been associated with reductions in the biological activity and or potency of the drug. They can also affect product stability by degrading the active pharmaceutical ingredient and or excipients and shorten the product's shelf life. And of course, last but not least, they can affect product immunogenicity either by acting as adjuvants or by inducing immune responses to themselves. And all these effects are not necessarily independent. So the immunogenicity risk associated with synthetic follow-on peptide products can generally be evaluated without any clinical data, based solely on an extensive characterization of the follow-on product, which aims to demonstrate the sameness of the active pharmaceutical ingredient and the comparability of the impurities found in the follow-on with the reference-listed drug. And for peptides that are considered of higher immunogenicity risk, as was extensively discussed yesterday, extensive comparative immunogenicity assessments leveraging assays such as innate immune assays and adaptive immune assays can suffice. In some more rare cases, clinical studies will be considered necessary for a synthetic follow-on peptide. Now, for follow-on peptides made recombinantly, typically to date, those have been evaluated for immunogenicity risk using clinical study data, simply because these recombinant peptides, as you can imagine, as was mentioned, have been considered a little more complex and less clean than synthetic products. The question we are posing today is, can advances in vitro, in silico methods and analytical methods support use of non-clinical data or reliance on non-clinical data, as has been done for synthetic follow-on products. The ICH Q6B guideline that layouts test procedures and acceptance criteria for biological products list O-cell proteins as process-related impurities to be minimized by use of appropriate, well-controlled manufacturing process. So that's about it. There's not much there. So in practice, how have manufacturers dealt with this? So I will start by saying that O-cell proteins present a few challenges compared to other critical quality attributes. The first one is that the mixture of O-cell proteins that you're going to get in a drug product is specific to the expression system and the process, which means that it wouldn't be practical to really compare the O-cell protein profile of your follow-on product with that of the product that it references. And the second one is that not all O-cell proteins are created equal in terms of risk. Some will pose a greater risk to patients or to product quality than others at different levels. So drug manufacturers typically use a combination of orthogonal methods. What is typically done to quantify total O-cell proteins in the drug product at different stages of the manufacturing process is running ELISAs. And these ELISAs usually use polyclonal antibodies that are produced by an animal immunized to the cell, to the expression system. The main challenge with this is that of maximizing the coverage of O-cell proteins, knowing that, of course, if you immunize the animal will not produce antibodies to the whole spectrum of O-cell proteins. And, but this will give you an idea of your total content in O-cell proteins and will not tell you the nature of the O-cell proteins present in the drug product. So usually, increasingly so, well, at least there's a lot of talk about using mass spectrometry, which is a high throughput method that has good sensitivity, but also has its challenges, notably in the interpretation of the result, and also requires, I think, instruments that have a big dynamic range, just because O-cell proteins are present in the drug product at much lower levels than the peptide product itself. And once you've identified O-cell proteins, you can estimate or evaluate the risk associated with those O-cell proteins. Typically, a manufacturer will do this using the published literature. If you can determine that the O-cell protein has a specific biological activity, and using their own experience. And based on risk, of course, these will direct the control strategies needed, whether it's reducing the level of O-cell protein or eliminating it. Now, there are knowledge gaps around this. So manufacturers typically target an upper O-cell protein limit that is laid out in a USP general chapter about residual O-cell proteins. And that limit, you probably all know it, is 100 parts per million. This threshold is driven by manufacturing capability and not really by clinical risk. Because as I was saying, a lot of these follow-on recombinant peptide products, or even in general recombinant peptide products, have been approved through clinical studies. So there has been no real need to further explore the clinical risk associated with these O-cell proteins. However, we are here to discuss this. We think that advances in technology may support the use of IVISIA to assess the risk associated with O-cell proteins. So we decided to, we formed a group and we decided to investigate and learn from the public what manufacturers do, because we know that as part of the purification process, right, there's a lot of cell proteins are controlled and evaluated. So we issued a federal notice back in July. I have to say that the comment period is closed, unfortunately, but we still hope to get more comments. So we are in the process of reopening it. It will take a few weeks, like everything we do at FDA. In the meantime, you can still mail comments, and I can give you an address. But again, it will be reopened because we want to collect more information. And specifically, we're interested in the methods that are used to detect, identify, quantify host cell proteins. We are interested in the minimal residual amounts that can be achieved, because we think that a lot of manufacturers could go down much below the upper limit of 100 parts per million. And we are interested in how the risk associated with these host cell proteins is characterized. So the first question has to do with the levels of host cell proteins. The second has to do with the challenges in reducing host cell protein levels. The third has to do with the analytical methods used. So we specifically name ELISA and mass spectrometry here. And the fourth, again, has to do with the technical challenges of running ELISA and other quantitation assays. And questions 5 and 6 are probably the most relevant to this crowd. And I believe that the next speaker, Vibha Jawa, will help address some of these questions. So question 5, are there qualitative or quantitative characteristics of host cell proteins that are associated with a higher likelihood of a clinical adverse event? And what tools do you use or plan to use to, again, compare the immunogenicity profiles of two products with different host cell protein profiles? And I will end by thanking all the members of the working group that I lead on the topic, a lot of whom are here today and have spoken, organized, and as well as Steve, who introduced the session today, who chairs the Immunogenicity Review Committee in CEDAR. Thank you.
Thank you, Sophie, for this great overview, summary, and the explanation on what's the state of the conversations that are happening in the agency to better understand these aspects of the host cell proteins and their analysis and mitigation. So the next speaker for this session is Dr. Vibha Jawa. She doesn't need much introduction, but I'm going to say that she's the Executive Director in Clinical Pharmacology, Pharmacometrics, Disposition and Bioanalysis with the Discovery and Translational Science Organization at Bristol Myers Squibb. So Dr. Jawa has an enormous amount of experience in bioanalytics, and she has been looking at different product quality attributes and how that impacts in the safety of the product, and also in particularly in immunogenicity. Today, she's going to present their work in the risk assessment of host cell proteins related to therapeutic proteins and cell gene therapies. This is studies that they are conducting within the programs that BMS has in gene cell therapy. Welcome, Dr. Jawa.
Everyone, thank you so much, Montserrat. So thanks for the introduction. So I can go into the next slide and talk to you more about the outline today. I'm going to speak about cell and gene therapies because that's what I'm currently doing, where we have a lot of questions, and we would like to contribute to this, the register of the notice, because we missed it when it was open. So thank you for opening it again. But I'm also sharing with you some of the work that we have done from my time at Amgen, because that's where we started thinking about critical quality attributes as more from a perspective of how to reduce and mitigate and do less in clinic, and also in a way reduce the all the burden that we put on the patients to collect a lot of samples to analyze the safety and immunogenicity risks. And so then I'll move forward, and then I haven't added slides from the time I was at Merck, but we did do a lot of work there on peptides and some unnatural amino acids in the peptides that we were working on, because I wasn't sure of the time here. So I can quickly summarize those, but we are publishing those, so they will be out soon. And then I'll end with some of our recent work, which was also published on the residuals associated with cell and gene therapies. So this slide is redundant. I think everyone knows what the immune system does when it sees danger signals. And we have to be aware of both red flags in our product from a perspective of host cell proteins for early innate phase as well as the adaptive phase. And so any functional assays that we are trying to build should incorporate all these mechanisms and include the cells that I'm showing here in the pictures. What we do very well, which we have also worked with the regulators both here in US and also globally, is how to look for anti-drug antibodies. And that's something which is a given when we start doing clinical trials. But what we do not do necessarily for biologics is to look for any kind of cellular responses, because we have in a way established that our fully human antibody-based proteins are they are not necessarily going to trigger a very effective T-cell dependent immune response. But then our experiences have told us that it's not the case, because even though the intent is to make a fully human protein, we are always going to have to worry about post-translational modifications, or we may have sequences in the CDR region which are going to be non-self, and they are going to trigger a T-cell dependent antibody response, and that's how we detect those antibodies. So even though we're not going to look for involved cellular immune responses, which are CD4 T-cell driven in the clinical subjects or samples from clinical trials, we could at least remove those drivers of those CD4 T-cell dependent antibody response up front, and that's where the mitigation strategy kicks in. So why were we interested in the innate immune system? And I'm going to quote the papers that we have followed, Daniela's work, quite a bit. And it was a good way for us to know what the agency was thinking, what was in their minds when we started designing or devising our strategy for looking at the risks upfront, as well as how to mitigate them. So these are two key papers that while I was at Amgen, we followed quite a bit, and it was about the low levels. And we would always think that, yes, the levels are low, so why are we so worried? Let's purify them out, and we should be okay. But that may not be the case, because if we are going to go into high dose of a recombinant protein as a dose, which we all know we are always striving to increase that formulation to be high dose, you're also increasing the amount of these impurities that you're going to deliver to the patients who are, again, not immune robust. They may be hyperimmune, like autoreactive, or they may be oncology patients, so there you're also giving them something which they cannot afford to have. So irrespective of whether it is low level, but it could be a high level when you talk about a dose. And additionally, even a low level can break the tolerance. And now, after 10 years since when this paper was published, we have seen that in our recent examples where even a combination biologic, when it's given with another immunomodulatory protein, it can break the tolerance of that first biologic, which could have been silent or inert based on our sequence-based risk assessments. So the human body can break tolerance by a sudden trigger. It could be dose, it could be just in combination with something else which they're being administered. So as much as possible, if we can reduce these risks, there is less likelihood of seeing those kind of events occur in the clinic. So now another aspect of Daniela's paper, which we followed, was her ability to look at all the mechanisms in the innate responses. And it was very elegantly looked at in the terms of which red flag or which impurity can cause what kind of innate phase responses to become more profound. So I've listed some of them in terms of the pathogen-associated molecular pattern receptors. And we also saw that there was a lot of characteristics of the PAMP receptors that could work in different disease states, or even the way we administer these drugs, whether it's a sub-Q environment or IV, or even like targeted tissue-based environment that we are seeing now with our newer drugs. So some examples, this is not just hypothetical. We have seen impurities and contaminants. And today we are talking about host cell proteins because that's the topic of my talk. And I know I said impurities in general, but we will go now and focus on host cell proteins. So this is a review article that I published with Dr. Wen from Eli Lilly. And we did a comprehensive look at all the critical quality attributes from both preclinical and clinical studies that we thought was attributed or reported in the literature to some kind of adverse or a clinical sequelae. So I'm giving some examples of the products that we found. And you can see lebrikizumab, which is my case study for today, with also an Amgen drug product with similar HCPs. We're going to delve deeper on it, but overall, we found that there were impurities and contaminants, host cell proteins, which could be either E. coli related or Chinese hamster ovary related, as well as microbial contaminants that could act as agonists and cause enhancement of the immune responses. So some of the clinical or the preclinical readouts that we captured from the different literature are listed, and it varies. It varies from being able to find actual anti-host cell protein antibodies in people. And then there were also some level of hypersensitivity reactions, local inflammation, which could be resolved, but it was still there. Now, to attribute something to a host cell protein, that this is an adverse event caused by that impurity is very difficult in clinic because you're seeing the entirety of the response to the rest of the product too. So unless we do some kind of experimental clinical trial in people, which we can't, we're never going to be administering something which is impure into people. So you have to rely on preclinical studies, which may or may not translate. But I think for innate responses, there is a good ability for us to understand if the TLRs are engaged or any of the other pattern recognition receptors are engaged, because there is a homology, which Daniela mentioned yesterday, even between non-human primates and humans. So definitely with these kind of studies we collected, we did find that there was some correlation on what people were seeing and then in preclinical and then what the response was in the clinical. So I'll go now through a few case studies. So this is the one which was published again a while ago, and it was about the GM-CSF-based products, and both of them were E. coli. They were produced in E. coli and showed higher immunogenicity. But interestingly, the specificity in the trials was quite different. So the GM-CSF product, we'll call it X, had a proline residue at the N-terminus compared to its human counterpart, while product Y was completely like a human GM-CSF. However, in the clinical trial experience, the product X with the proline residue, they did detect immunogenicity in the serum, which was specific to GM-CSF, and they also had neutralizing antibodies that impacted the efficacy. For product Y, the ADAs were seen, so it was GM-CSF was homologous there, so it was very mild and non-neutralizing. So the self protein actually helped to reduce that immunogenicity risk. However, patients receiving the product Y developed antibody responses to the E. coli host cell protein. There were two E. coli host cell proteins that did have an immune response. The weak ADA response to GM-CSF was good because that was their drug product, but then they had to deal with this ADA response to the host cell proteins, which could have, and the explanation there was that because GM-CSF is an immune enhancer, that could have enhanced the immunogenicity to those host cell proteins. So irrespective, you can see the interaction of host cell protein, which may be even be present in small amounts, but just because you have a mechanistic interaction of your product with the immune cells, you may exacerbate that response. So a good learning from this that you still need to eliminate, even though if you have a homologous product, the HCPs are still going to make it more immunogenic or cause additional immunogenicity to other proteins. So here are some examples. This was a slide provided by EpiVax. Thank you. So this just shows in our biologics, our peptides, we keep on observing like clinical trial news about halting of trials or adverse events or formation of anti-HCP antibodies. So this is a big burden for all of us because if we go into a clinical trial and start finding these challenges, we have already spent 7 to 8 years in developing a drug, which is now going to be either halted or has a pause, and you have to go back and resolve the issue before you can administer or continue with your trial. So that's a big limitation for any, if you have experience, it's a lot of stress in the company, and bioanalysis folks like us are the first ones to be blamed that, oh, your assay did not detect or you detected too much. So it's always the blame comes back on the preclinical development folks that how come you did not look at this up front and how come this was not controlled. So clinical trial experience shows us that these are things that should be mitigated upfront and not wait till we first see it in clinic because it just is a baggage that you carry over then. So now I'm going to go to a lot of work that was done at Amgen on our own drug products. And we used to, Amgen has a IgG2 monoclonal antibody-based platform that is made with xeno mouse that they acquired. So this mouse system makes human antibodies. But prior to that, we also use Chinese hamster ovary-based expression systems. And Chinese hamster ovary is like a mammalian cell line, which is very good at expressing, but the limitation is that whenever you elute or whenever you purify these protein antibodies, you're going to co-elute some amount of the host cell protein with it. You can do as much purification as you want. There is an uncertainty with the co-eluted peptides that come with the drug product. And the only thing people can do is keep on running a lot of factor 8 column, not factor 8, FVIP columns and HPLCs over and over, but you are just adding more and more steps, but these don't go away. So you will be left with some amount of parts per million of these HCPs at the end of the final formulated product. So when we were developing at Amgen a drug product, and we found that between the phase 1 material or even the IND-enabling GLP tox material to the phase 1 to the phase 2, we started noticing that our host cell proteins from Chinese hamster ovary were fluctuating. They were sometimes 40 ppm, they became 140 at some point, then they increased to a larger number, and everyone was alarmed and said, go back, purify more. So in the meantime, as a clinical immunology group at Amgen, which was focused on understanding the immunogenicity, we were asked, what do you think if the host cell proteins go from 40 to 160? Is that a risk for the program? Which otherwise was a very good, like, solid antibody, and they were worried. It was, it's the published product now, it's, commercially approved product, adalimumab, which is the migraine medication, which is doing very well. So I think the first thing we was like instinctively was let's develop an anti-HCP antibody assay, and we are done. We can go into clinical trial, look for anti-Chinese hamster ovary antibodies and give that data to the agency. But that's not going to solve the problem. The people are going to still have a certain risk. So we looked around, and Genentech had done a very extensive analysis with their program, lebrikizumab, and they, in fact, had gone ahead and developed an immune ATA assay to one of the component, one of the HCPs called the PLBL2. And of course, they found 90% of their clinical trial subjects came up positive for these antibodies. So that does not help, because what are these antibodies doing apart from their own immunogenicity assessment to lebrikizumab, which was minimal and which was not very impactful on the PK. So they also knew how much of that, the drug products had how much of that PLBL2. They found that the PLBL2 antibodies increased over time, but it did not seem to impact the safety or immunogenicity or even the exposures. It did not have any immunostimulatory activity. So Genentech's solution was they reduced in their future products, which went into the phase 3 pivotal, they reduced the amount of PLBL2 by doing a lot more purification process. But Amgen did not want to do that. We were already in phase 1. We had all this data, and we asked, like, is there another way we could approach this? So we looked at 334, which was the arenimumab, and we found that the sequence had next to no risk from an intrinsic risk perspective, and we did not also have much liabilities from any post-translational modification. So again, a great molecule. However, when we look at, we wanted to ask the question about host cell proteins in general, not just PLBL2, even though we had started noticing in our profiles that PLBL2 was one of the higher eluted proteins, which was coming out with the drug product. So when we put this into the ISPRI tool, at that time, we would, Annie used to call it choppy. So we were using ISPRI already, and she said, use Chinese hamster ovary-derived informatics tool, because we have developed it just for looking at this. And it followed the same concept, looking at the sequence, looking at its ability to bind the pockets, and then, but it was looking at similarity to the mammalian genome, especially the Chinese hamster ovary genome. And as you notice, the PLBL2 antibodies were in 90% of subjects. So somehow we are exposed to these kind of similar mammalian proteins, and we have a pre-existing reactivity sometimes to these kind of proteins. So, and there is a lot of homology as a mammalian protein with these CHO proteins, but then there are also some differences. So if we could look at the host cell proteins and understand where those differences are, we did not have to worry about everything which came out of that HCP co-eluted fraction. We only had to worry about specific ones and then try to mitigate those. So again, this is a paper that Genentech published. It's a very well-cited paper. I would encourage everyone to read. Sally Fisher was the first author. And then this was something that Genentech published later on, and it said that it co-purifies, PLBL2 co-purifies with most of the large molecule antibody-based therapeutics, and it is one of their most usual suspect when they're worried about HCP. So I mentioned about Amgen work, so we also published it later, but we went with it in a very different way. We said, we're going to look at host cell protein for our program, the one which was under scrutiny, but we wanted to do it as a platform approach. And as I mentioned, we were making antibodies in a very similar way. So we said, we'll take 4 to 6 different antibodies that Amgen had made. For some of them, we already had clinical experience, so we had the drug lots and the amount of HCPs in them. But they were also different because some of them were against metabolic targets, so we knew they're not going to be immune stimulatory, but the other ones were against immune modulating targets. So we knew they could increase mechanistically, like the GM-CSF, they could increase the immune responsiveness. And then if the HCP is present, are we going to, am I off time? I'll rush. So, so then we took 4 to 5 different antibodies, and we asked our CMC colleagues, can you tell us the lot-based HCP distribution for all of them? And then that was all the LCMS and the ELISA data that they had already collected for their CMC package. So nothing new was generated. We were leveraging what was already there. We did find, and I'll go to the next slide because this I will skip this because everyone knows the methods. So this is something that we did first. We found that when we looked at all the antibodies that Amgen had made, there were some usual suspects. There were 24 host cell proteins from Chinese hamster ovary, which would always be eluded. They were at different levels. And so we used the CHOPY tool or the ISPRI tool and rank ordered them for their risks, for the scores of the T cell epitopes. And in the CHOPY, we had the homology to the Chinese hamster protein. There was a Janus matrix at that time, but it was in the pilot stage. So we also used that to see how much of these proteins have homology to us in terms of us not reacting to it. So overall, the rank ordering was done based on probability of the number of non-self T cell epitopes and also finding that which one was more self or non-self. Then after that, we looked at the amount of actual concentration based on the LCMS data. So what we ended up doing, and how do I go back? Okay, so this is how we scored it in the sense that we knew that if you are on the top right corner or you're in the top left corner, you are the highest risk. And in the bottom right corner, you're at the lowest risk. So all those proteins were separated based on this kind of a quadrant plot. So when this on the left is the tool, and this is the paper I'm talking about. But overall, PLBL2 was still coming up as a problematic. And we were worried about it, and we said, okay, let's see how much we can remove it, but still we were left with quite a bit in the overall ppms. So this is the study I mentioned. So what we did was we took 4 different drug products. We took the in vitro human PBMC assay that we had developed in-house and asked the question, let's challenge these cells, collect the initial data for innate response based cytokines, and then follow the same product forward and then look for adaptive phase immune responses. And so in a way, we kept the culture the same. We normalized whatever cells we added in the front and continued with that. And then if you look at the profiles, it's very interesting. It just shows you the intrinsic proteins that are associated with intrinsic immunogenicity that is associated with each of these drug products. And then the darker shade is one where we have the HCPs in it. So one of the products is the final formulated product where we have done the best. This is your best bet for removing the host cell proteins. They still have some which range from 40 ppm or 100 ppm. But then instead of using, we use other controls that we are calling suitability controls and all for our assays, we asked our CMC colleagues, our manufacturing colleague, can you give us a 2-step before process-derived material which we have not purified? We would like to see how that looks in our assay. So the bottom line was we could increase the threshold from what was analytically mandated threshold, which was around 100 ppm, to almost for some molecules to 4,000 ppm and not see a change. And good part was some of these products had gone into clinic in terms of not that high, but we knew that if they ranged between 100 to 160, that was not changing the clinical risk of our of the subjects. So it was very obvious that analytically you could detect even 13 ppms of these proteins, but when you go to biological relevance, they were not very relevant, and the in vitro assay showed that they overlapped quite a bit with each other, whether you had low amount of ppm or high. But if you look at each profile, it looks very different because that's the intrinsic immunogenicity of each molecule itself. What did they contain from an in silico perspective? So I think I made my case. Now, this is data that Epivax generated to show that whatever they predicted as in silico is also showing up. If you just take proteins in isolation, you are seeing that they also proliferate a lot of T cells compared to the other negative controls. So in a way, the predictive in silico was telling us that, yes, this protein is a problem, try to reduce it or try to mitigate it by other means. So this is another example. I won't go here because this is showing that some of the rabbit data can also be very correlative to what you would see in clinic. And so I want to point out to the rabbit intravitreal co-administration of some host cell proteins with ranibizumab. It triggered local inflammation, and then this was something which was also observed in the clinic. So again, for innate responses, there is ability to understand some of these risks due to host cell proteins up front. Now, novel modalities, I'll go very quickly through it. So we are developing both cell and gene therapies. AAV gene therapy, we know there are contaminants or residuals that are concerning empty capsids. So I won't share that data today because we terminated that program, but our CAR T cells are using CRISPR or AAV and other vehicles to transduce genes into the engineered domains of CAR T. And then we are always worried that you're going to express those when the CAR T cells go into the patient. So what we did here was our questions were related to AAV-induced gene that was helping to reduce or to enhance the cell expansion, and CRISPR to knock out the HLA class 1, because we are class 1, yes. So we were looking for, are there residuals of AAV and Cas that we need to worry about, which can impact the expansion of the CAR T cells when the entire domain is expressed. So we were almost ready for trial. There is no preclinical stage in CAR T programs. It goes from discovery directly into clinical. And so we didn't have any data to tell us what that risk would be. So we decided we're going to take AAV and Cas at different doses and do an in vitro and an in silico assessment and generate that data, go to the agency with that pre-IND package and submit it and say, we are going to look for antibodies to our ECD domain, which is a given, but we are not going to look for antibodies to AAV or Cas, and we're not going to do CTL responses to AAV and Cas. And the reason was not because PMS doesn't want to do it. The reason is the patients for CAR T trials are extremely sick. They are at last line of treatment. They have no blood to give. They have already given blood to get re-engineered, and their cells are very fragile and apoptotic. So if you have to do any involved cellular assays, the results will be inconclusive. And we had already had that experience. So we made the case, we gave the data to say the risk is very low or none. And this paper is very good to read. It has some level of thresholds incorporated in it, which we also looked at. So definitely please read it because I don't have time right now. So, but my summary is that host cell proteins, we know they are red flags. They can even pose risk even at low levels. We can do a lot to change our processes, but sometimes doing an effective risk assessment, which relates directly to what would happen in clinic, could help you to mitigate that upfront rather than taking something forward and like finding it in a surprising way. The control specifications and thresholds can be made a little bit loose and not be based on analytical, they should be based on biological responses. And monitoring of immunogenicity, and this is what we are trying to tell all of our teams, let's look at the lot-to-lot changes. Let's collect all the safety information and ask, are there changes in the lots, HCPs from lot to lot, which we can then capture and say this is attributed to a change. And of course, novel modalities, as we go along, we're finding more and more engineered variants that we will need to work as impurities or call them out as impurities. Thank you so much.
Thank you, Dr. Jawa, for such a broad overview on HCPs and very informative case studies. So our next speaker is Dr. Emilee Knowlton. She's a senior immunology sales specialist at ProImmune. She's interested in providing innovative solutions for clients that radically improve the understanding of desired or unwanted immune responses. Today, Dr. Knowlton is going to talk about introduction to MHC-associated peptide proteomics. We know these assays as maps for characterization of presented peptide epitopes. So please.
Okay, well, thank you for that introduction. And also thank you to the organizers for the opportunity to share ProImmune's experience with the MAPS assay, which again stands for MHC-associated peptide proteomics. Now, MAPS assays are such an important and commonly used tool for assessing therapeutic proteins that it almost sounds strange to consider it as an alternative approach or an alternative method, but it's certainly an underutilized tool in the generic peptide space, especially given the success that companies have had in using MAPS to characterize their biologics. So today I'm going to be introducing the assay as a potential option for understanding immunogenicity risk of generic peptides. I'll be going over the methods as well as various applications, and also providing some case studies from the ProImmune version of the MAPS assay, which is called ProPresent. And I'll also touch upon some considerations for experimental design and implementing MAPS into those assessments. So MAPS assays are highly sensitive and very robust in vitro assays that can be used to evaluate immunogenicity risk of a protein therapeutic by identifying the naturally processed and presented epitopes from that protein. It's really considered the gold standard in the field for predicting CD4 T-cell epitopes and is a superior method to identifying peptide presentation compared to traditional overlapping peptide scanning approaches. It is a widely used platform by pharma and biotech industries to identify regions that are at risk for inducing unwanted responses and serving as potential candidates for either reengineering or de-risking or simply to be tracked in the clinical setting. Now, because the assay will only tell you about what peptides are being processed and presented and not how the T cells will then respond against those sequences, it is often used in combination with T cell proliferation assays to confirm the functional relevance of those presented peptides. And then it is quite a versatile assay. It's been used in a number of different applications to address different questions, including ranking your protein candidates by promiscuity of those presented peptide epitopes, to assess deimmunized protein sequences compared to the parental sequence, to identify peptides from host cell proteins, to evaluate the impact that protein modifications can have on that peptide presentation profile, and then also to investigate the drug's mechanism of action. So the methods for the MAPS assay is shown here. As mentioned yesterday, you want to work with a broad panel of HLA-type donors so you can have a good global representation of various alleles. We work with cryopreserved PBMCs in this assay to generate monocyte-derived dendritic cells, which serve as your APCs. Those cells can then be loaded with the protein of interest to allow for antigen processing. You then mature the cells, which upregulates class II on the surface with those peptide fragments bound. Following a lysis step, you can do an immunoprecipitation to pull down those MHC peptide complexes using an antibody against either HLA-DR, DP, or DQ. You then recover the peptides and sequence them using a high-resolution sequencing mass spec, or LCM-SMS. You then generate a database that contains the human proteome as well as the sequence from your input protein. And you essentially probe that database with all the mass spec peptides that were obtained to determine what was presented from your protein. Now, in addition to this kind of classical class II approach, we can also look for presentation via class I, and this is typically in the context of antigen target identification for vaccines and immunotherapies. But because the dendritic cells used in this assay are not great at cross-presenting exogenously loaded material onto MHC class I, we do have to alter the method slightly. So what we do is our clients actually provide us with antigen-experienced cells, so something like a virally infected cell. We then do a pan class I pull down and recover those peptides and sequence them as I've just described. So as I mentioned, the ProImmune version of the MAPS assay is called ProPresent, and we really have an unrivaled client publication record with this assay, and some of those key publications are listed here on the right. Our customers have used ProPresent to interrogate a wide array of input material, which has been characterized here in this table. And of course, we can't share all the details, but just as a few examples, we have worked with monoclonal antibodies, viruses, whether that's viral vectors, individual viral proteins, or even whole viruses, cancer antigens, model antigens, ADCs and bispecifics, as well as pegylated proteins and other alternative scaffolds. And you can see highlighted, we have also done some work in the generic peptide space, but don't get too excited, I won't be able to share any of that today. So before I get into the case studies, here's just an example of how the data is provided to you. So you are given the donor's HLA type, as well as any sequence that they've presented from that protein of interest. And then each sequence has an expect value, which is indicative of peptide identity and is a measure of the predicted mass of the peptide versus the mass spec measured mass. So as an example, donor number 11 expresses DRB1 0301 and 0401. They presented 2 unique peptides, one from the light chain and one from the heavy chain. And you can see within that heavy chain region between amino acids 77 and 93, there's actually several individual fragments presented. So when you look at your cohort collectively, that's when you can start pinpointing those promiscuous sequences or those that are presented by multiple donors with different HLA types. And these are really the ones that are at higher risk for inducing unwanted immune responses due to that broad population coverage. So as an example, that DNA KN sequence is actually presented by each of these donors expressing those different alleles. So these are certainly peptides you'd want to consider to take into the functional T-cell proliferation assays. So we have 3 case studies to share that really highlight the versatility of a MAPS assay, as well as how we've helped to address different questions asked by our customers. And this first example comes from Editas and the FDA, and it's the use of ProPresent to characterize CRISPR-Cas9 immunogenicity, which again, as we've just heard, CRISPR-Cas9 is a genome editing tool, but Cas9 is of bacterial origin. So having any responses against that bacterial protein could have serious implications. And so we performed an 18-donor ProPresent study, and the alleles of the individuals used in the study are shown on the y-axis in the far left. So to give you an idea of the HLA coverage of this size study, this is about 70% of the North American population. Now, each different color represents a different donor. So you can see that multiple donors are presenting the same peptide across this Staphylococcus aureus Cas9. And overall, there were 22 unique peptides that were not only identified by ProPresent, but also resulted in T-cell proliferation, which is the pink data set that you see overlaid on top of the ProPresent data. So these 22 sequences represent about 85% of the sequences we detected by mass spec, and then each individual presented between 2 and 10 unique sequences. So this is a really nice example of how you can get very detailed characterization with this type of assay. Okay, the second case study, if I could progress here. Okay. This is a really nice example of a question, a very pressing question that we're commonly asked, and that is how predictive are these tools in reflecting clinical outcome? And of course, as we heard yesterday, there are multiple factors that lead to clinical immunogenicity, and we're looking at risk assessment. But this is a really nice example of assessing sequence-modified drugs. So this is a retrospective study performed by Novo Nordisk, with Novo Nordisk and the FDA, looking at Vitreptocog alpha, which is an engineered factor 7A analog that had to be pulled from the phase 3 trial when there were ADAs observed in some of those patients. Now, Vitreptocog alpha just has 3 amino acid substitutions compared to the native form that's been used for more than 20 years with no issue. And so this group wanted to use both the MAPS approach as well as T-cell proliferation assays to see if they could essentially predict whether these neoepitopes had been engineered in. So there were 2 independent studies performed, one MAPS assay at Novo Nordisk on 17 donors, and then we did a 12-donor ProPresent study to look at presentation from a recombinant factor 7A as well as Vitreptocog alpha. And you can see there in the protease domain where those 3 amino acid substitutions were incorporated. So a V to D, an E to V, and an M to Q. And all the mass spec peptides that were attained in both studies occurred over those V and Q mutations. So they did go also on to do the T-cell proliferation assays to show that there were CD4 proliferative responses that were enhanced in the V and Q mutations compared to those native sequences. So even though there were only 3 amino acid substitutions incorporated, 2 of them were processed, presented, and resulted in T-cell activity. So it was determined that immunogenicity was the cause of failure for this drug, which these assays were able to reflect. The third case study also helps to address a couple of very important questions pertaining to MAPS. One, which was discussed yesterday, and that is, why do we use healthy donors in this type of study? So this is a project with CSL Behring and the FDA. in which we were looking at presentation from a full-length recombinant factor VIII, as well as a platelet-derived factor VIII in the presence of von Willebrand factor. And not only did we perform this study with healthy donors, which is shown in those blue peaks, but we also looked at hemophilia patients. And what you can see is the number of peptides or the magnitude of response across factor VIII was higher in the healthy donors, but the peptide presentation profile is nearly identical between the two cohorts for both proteins. Now, in part D, another question we're asked is which loci to focus on. And obviously, HLA-DR is better characterized, and most groups will focus on DR-presented peptides. But this is a nice example of why you want to perhaps look at all three loci. So this is the presentation of peptides from factor VIII, and it's quite a large protein. And you typically see a large number of peptides presented from large proteins. So as expected, you see nearly 1,800 that are restricted to DR, but in summation, you see a total of 500 between DP and DQ, which are potentially immunogenic sequences that you could otherwise miss if you didn't look at this. So given the robustness and sensitivity of MAPS with demonstrated success in other research areas, the assay really holds untapped potential for assessing immunogenicity risk for generic peptides. Specifically, MAPS could be applied to the statement we see here from the ANDA industry guidance stating that such data should demonstrate for each new impurity that the impurity does not contain sequences that have increased affinity for MHC. And we heard about addressing this yesterday using T-cell proliferation assays. But of course, MAPS could serve as an orthogonal approach to understanding if those impurities are actually processed and presented. And because you are using a broad panel of HLA-type donors in this assay, you'll be able to incorporate a diverse array of MHC alleles, which is another important component to addressing this question. And if those impurities are in fact processed and presented, you could go on to determine the precise either MHC restriction or affinity using physical peptide binding assays. So again, the objective for using MAPS in the context of assessing generic peptides would be to determine whether those impurities get processed and presented. Does not like me. Okay, so over the next few slides, I have just a few experimental design considerations and implementing MAPS into those assessments. And the first focuses on how to go about testing those impurities. And we heard a little bit about this approach from Eli Lilly's presentation. And I'm going to use semaglutide as an example. You can see that there's a large number of potential impurities, different truncations, insertions, deletions. And the MAPS assay is quite a cell-intensive tool. So you are limited to the number of test articles that you can assess over any given cohort. And so you wouldn't be able to test each one of these impurities individually, but would rather have to use a pooled approach. And by pooling samples together, there are some inherent advantages and disadvantages. You can, of course, incorporate that element of competition so that you see that the stronger epitopes are being presented. But this also means that you could miss some presentation of other impurities that would be done so, albeit at a lower amount, because this is a non-quantitative assay. So the impurities are either going to get processed and presented or they're not. So certainly something to keep in mind. Now, the quality of impurity has already been discussed a lot yesterday. So I'll just mention the purity of the impurity is something to keep in mind. And then lastly, the concentration in which you are testing these impurities is quite important. So this assay is typically performed in molar excess. So we're really overloading the system with proteins so that we can see presentation. So we might need to think about the strategy differently and use an appropriate concentration to still address the presentation question, but be at physiologically relevant levels. Okay. Another consideration is how you go about generating that database. So this is likely going to be much more tailored compared to just putting in one protein sequence. You must know the sequence of each one of the impurities that you're searching for and would have to input each specific insertion, deletion, modification, for example. You can also search for mass changes using this, so this is really a key advantage, as well as non-natural amino acids. But we do know that modifications can be somewhat challenging to search for. And we have good experience looking at things like phosphorylation, but stuff like glycosylation is quite challenging. So that's certainly something to keep in mind as well as a potential limitation. And then finally, controls have been discussed throughout this entire workshop, and the MAPS assay is no exception. We work with endogenous reference proteins in the ProPresent assay. So we like to see peptides presented from at least a subset of the proteins that you see listed here so that we have confidence that the cells are behaving as they should. So we search the human proteome element of the database. Other groups use things like tracer proteins that have been added into the assay or test antibodies with known germline sequences. But again, we may have to consider other controls that are more relevant to assessing these shorter peptides versus what we would typically do with a large protein. And then there are, of course, a lot of variables in how the assay is actually performed, whether that's in-house or across various CROs that could impact how these samples are processed and presented. So this includes things like the cell culture, how the DCs are generated and matured, the time that you allow for the antigen processing, the antibodies that you use to pull down the MHC peptide complex, as well as how those peptides are recovered. So optimizing each one of these steps, could enable better assessment of these impurities potentially. And then I've already listed a few of these limitations, including the large amount of cells that are required to run the assay, the fact that it is non-quantitative and that you are limited to which modifications that you can search for, and then you're always going to be limited to the sensitivity of the instrumentation that you actually have at your disposal. So I'll just leave the group with some more considerations before the coffee break. The MAPS assay is a research service. It's performed as a research assay. So we need to think about how we would go about validating this assay, as well as demonstrating the sensitivity of MAPS. Should it be used in combination with other assays like physical binding assays to look at either the MHC restriction or competition against other epitopes? We know these impurities are going to be at low concentration, so are we going to be able to see them being presented from the API, or are we just going to be able to see it when it's spiked in as either an individual impurity or as a pool? And then finally, we know that we see a large peptide presentation profile for large proteins. So what's that going to look like for shorter sequences that are less than 40 amino acids? Okay. So by, again, optimizing some of these approaches and also honing in on some of these details, we may find that MAPS really holds untapped potential for assessing generic peptides as an orthogonal approach. We know the MAPS is a very robust and sensitive and very versatile tool that's been used successfully to characterize a wide range of input. And at ProImmune, we have decades of experience in providing these best-in-class immunogenicity risk assessment assays, including ProPresent. And this specific tool reports in about 6 weeks, which is really nice for accommodating these very tight deadlines in this competitive market. So as Jeremy mentioned yesterday, we do work very closely with our clients to support regulatory success, as demonstrated by the success of our customers. So thank you very much.
Thank you, Emilee, for this clear presentation on the MAPS assay and the factors that to take into consideration and the applicability of these assays in understanding better the process of presentation of these impurities. We're about to start the second part of this session. The first talk in this part is going to be given by Dr. Howard. Dr. Kristina Howard is a research veterinary medical officer at the Division of Applied Research Science within the Office of Clinical Pharmacology in CDER at the FDA. Her research focuses in developing and improving in vitro and in vivo models to better predict adverse events associated with therapeutic proteins and immunogenicity risk assessment of the products. So today, Dr. Howard will present on an overview on alternative models to assess immunogenicity risk from humanized mice to microphysiological systems and beyond, and is going to focus on some examples related to genetic peptides and host cell proteins. Welcome to the podium.
Okay, thank you. Are you able to hear me? Okay. First, I'd like to thank the organizing committee for inviting me to speak today. And second, I'd like to thank everyone online for attendance. Good morning or good afternoon, depending on your time zone. So as Monsi indicated, I'm going to talk about other alternative models. So we heard this morning about the MAPS assay, which has not necessarily been traditionally used for generics. These two, I'm going to talk about two other types of models, but there are obviously even more models than that. So first, the natural FDA disclaimer that this is not policy or guidance. Okay, so I'd like to start with just this overview of different types of techniques. So there are many different potential approaches, and we spent much of the first day going over what would be considered in vitro approaches, whether it's adaptive immune approaches or innate immune approaches. However, there are other ways to look at things. We also heard about in silico, so different types of modeling techniques. But you can go further than that, and these are obviously going to be orthogonal methods, not necessarily the first method you might try. And one of the things we haven't heard a lot about at this conference is animal models, because you can't test immunogenicity in, for example, a normal mouse model, or typically even maybe a monkey, because if it is a truly humanized monoclonal or protein, it's going to be recognized as foreign and naturally be immunogenic, so the model would therefore be useless. And so we're going to talk a little bit about humanized mice, but I also want to introduce organ-on-a-chip culture systems. So in vitro systems really span a range. And so when you're looking, there we go. So we start off with 2D cultures, and this is really where what we talked about yesterday was 2D cultures and what we're most familiar with. However, you can then expand to something that's called an organoid, and most people use organoid and microphysiological system interchangeably, but they're not actually the same thing. And so this paper here that's referenced is from a former colleague from my division who worked exclusively on microphysiological systems. So you can start out with organoids, which can have multiple cell types. It is cultured, but you actually have multiple cells in there, so you're not looking just at an immune cell or just at a hepatocyte. And this isn't your typical Transwell plate type experiment that you think about. This is actually growing the tissue in culture. So you can do histological evaluation of them once you have a large enough size organoid, and you can test things in them. But obviously, this is still not going to give you the physiological structure of the tissue. So that's where MPS comes into play. And typically what MPS tries to do is, within a fabricated compartment, develop what the tissue might experience in vivo. And I have an example that I'll show you. And what is being attempted is individual systems for all of these, but also then connecting them together to try to model the entire body at once. And the one thing you don't see here is you don't see the immune system, because the immune system pretty much is everywhere. Yes, we have lymph nodes, yes, we have spleen, but the immune system occurs all throughout the body, and so it does make it difficult to incorporate. But that doesn't mean that this may not be a useful tool. So I'm going to use the lung as an example, because it's easy to visualize. So if we were to try to do a bronchoalveolar lavage, we would have to put a tube down, and we would have to have this go down and flush, collect the fluid, and then we would get the immune cells out of the lung, and we might be able to make some assessments. If you do this in the terms of an MPS system, on the one side, you have the airway epithelium, you have air. On the other side, you have endothelial cells in a membrane, and you can actually run air through the system and fluid through the other side of the system, so that you actually have something that more closely represents the tissue in the in vivo state. Now, obviously, this represents the lower airway. It doesn't give you the upper airway, meaning if you were to have a nasal vaccine or something like that, or a nasally administered generic, it's not going to get you the actual intake, but it does get you the lower lung. And there are lots of MPS systems. The one that I do want to point out is there are MPS lymph node systems where they're trying to develop an actual lymph node that exists in a dish where you have T and B cell interactions with antigen presenting cells. These thus far have been most successful with infectious agents as opposed to looking at immunogenicity, but these are ever evolving. And so as they become more evolved, these are things that may be useful. And I'm going to transition and talk about humanized mice. And so first I want to just point out the type of humanized mice that we use. There are many types on the market. Some of them have PBMCs. Some of them made are solely with CD34 hematopoietic stem cells. The kind that I make have thymus underneath the kidney capsule because you need thymus so that you have self-peptide present, and that way you can have proper training of your T cells. So the T cells recognize self, and that really becomes an issue. If they don't recognize self, they can't tell you if it's immunogenic or not. And we currently use, rather than stem cells that come from the liver, we actually use cord blood cells, and they're HLA matched. So we have HLA matching in our system, and these are implanted underneath the kidney capsule, and then following a dose of busulfan, we give the stem cells intravenously a couple weeks after surgery. And then we wait, and 12 to 16 weeks later, we get a human immune system inside of the mouse. It is not a perfect immune system, and it doesn't have everything completely reconstituted, but it has shown over the past number of years that it can demonstrate certain capacities of the immune system quite well. So I'm going to go over two studies first. These were with generic peptides, and fortunately, salmon calcitonin and teriparatide were very well explained yesterday by multiple speakers. So we had short studies that we did that were for 4 weeks, and we either gave saline, teriparatide, or calcitonin, and we gave these at different doses, so in micrograms per kilogram for the teriparatide, and for the calcitonin, it's international units per kilo. And these were sub-Q dosing. This other study, since calcitonin salmon can also be given intranasally, that study we administered intranasally to the mice, and that was daily. These were also daily, and this one went on for 8 weeks. Okay, so this is just the overview comparison. I'm not going to go over it since it was done very well yesterday. And these are some different readouts that we got from the mice. So all of these are splenic cells that we're looking at. So if we look at short-term memory B cells, in the teriparatide group, what you see is that the teriparatide-treated mice are no similar, they are not in any way different from the saline-treated mice. And if you look at the calcitonin-treated mice, what you see is an increasing trend of having more short-term memory B cells as compared to saline. So here they're about equal, but it goes up as the dose goes up. And short-term memory B cells are involved in primary responses. These are not the long-term memory B cells. But it does show that when you give calcitonin, there is a trend to recognizing something and more of them being present. These are long-term memory B cells, and we'll start the teriparatide. There is no difference between saline and teriparatide. And if you look at the calcitonin, again, the lowest dose is not different from saline, but we see this increasing trend again as we increase the dose. And the long-term memory B cells are the ones that are going to give you a greater secondary response. And I will say that for this particular study, we were unable to do anti-drug antibodies because we didn't have an assay worked out at that time. So I can't prove that these B cells are specific, but it's the only thing they got, and they basically live in a sterile isolator. So they really weren't seeing any other antigens. These are antibody-producing plasma cells. Now, these were in the bone marrow, and again, teriparatide, we really see no difference from saline. And with the calcitonin, we do see an increase in these cells. These are actual cell numbers that you're looking at, as opposed to a percentage. And the antibody-producing plasma cells that you see in the bone marrow are most typically IgG, as opposed to your IgM, which are the more transient initial ones. Now, if you recall, I said that we did dosing in the nasal cavity because that is how one version of salmon calcitonin is dosed. And what I'd like to point out, this is the calcitonin RLD. And you see these blue lines, that's showing the diameter of the nasal passage. Here's one impurity, here is another impurity. This is saline, and these are impurities that we were testing that were known to exist in a generic product. And what I want to point out here, number one, is you see the nasal passage is extremely small as compared to the others, and the blue arrows are pointing out all the immune cells that are present in the nasal passage. And we actually did some follow-up studies with this, and it appears it actually could potentially be an excipient effect that was causing it rather than the drug product itself. But what it does show is that the mice were able to respond to the inflammatory stimulus they were given. And with the impurities, you don't see anything, and with saline, you see nothing. And so this is one thing that having an actual animal model can give you that you can't get from a culture dish, and you really can't get from doing an in vitro assay. So again, this would be a limited type of use where you're looking at a specific product at a specific dosing regimen, but it would be an orthogonal method that if you had an issue, you might be able to follow up with it. So with respect to our generic peptide testing, when we treated intranasally, we had significant inflammation in the nasal cavity of the calcitonin-treated mice, but not the impurities or the saline. And when we looked at treating sub-Q, we found increases in B cells and antibody-producing cells to the salmon calcitonin, but not to the teriparatide. And this matches up very nicely with the data that was presented both in silico and in vitro with respect to these two products. And it matches what clinically occurs, which is the teriparatide is not immunogenic in humans, whereas the salmon calcitonin is. Okay, we also have looked at host cell proteins, and I don't have anything nearly as elegant as Vibha does, because this was more of a pilot study to try to understand whether or not the mice could respond to host cell proteins, because we felt since these peptides were so small, they're probably not going to do anything at all if we give them host cell proteins. So what we did in this study is we dosed mice for 8 weeks subcutaneously with either a positive control, which was a vaccine, and or a negative control, which in this case was saline. And we had 5 different host cell protein samples that were very courteously produced on our campus in our bioreactor and purified for us. And for one group, we gave them 10 nanograms per dose without cleaning up the DNA. And as many of you probably know, DNA is very immunogenic since it's going to bind toll-like receptors. And the other samples all had their DNA cleaned up, and they got 100 nanogram dose, and this was 3 times per week. And we collected a large number of parameters. I'm only going to show a few today due to time. And there we go. So the first thing I want to show is we use CD45RO as an activation marker in blood, and these are just looking at the histograms. These are representative histograms from the mice. And here we have our negative control where only 25% of the T cells have CD45RO. For our positive, it's about 56%. And then these are 3 different host cell protein samples. This is the 10 nanogram that still has the nucleic acids, and we see 45%. This is a different one at 100, no nucleic acids, and this one was 71. And this one also has no nucleic acids, and it was about 59. And what we were very surprised. Now, I will say not every donor responded, which is what you would expect with people. And when we do studies with these mice, we make sure we have multiple different donors of different HLA types so that you're getting different responses. And so some mice will respond very robustly and others did nothing, which is kind of what you would expect from a human population in a study. If you look at the number of naive mature B cells, so those that are able to respond, when they have saline, there's really not that very many there, but when you gave them anything, there were more. So they were at least sampling the antigens. It doesn't mean that they actually matured, but they were sampling the antigens that were being shown. And this is looking at plasma cells in the bone marrow. This is saline, where there's basically nothing Here we have our positive controlled 3%, and you can actually see very clear peaks here for the host cell proteins. This is one of them, another, and another. And they were all pretty robust. So for this particular study, what we did was we looked at antibody class switching. And this is all, of course, IgG class switching. So we'll start at the top with IgG1, and the asterisks are for statistical significance, meaning different from the other bars. So here, the saline's actually pretty high. And you do see some differences. For some of them, the IgG1 was lower. In other cases, it's relatively the same. We don't know if it's antigen specific. And if we then look at IgG2, you again see a different pattern. So the saline is a little bit higher than these two, but the one for host cell protein 8 is quite high for IgG2. This really isn't any different, and that's not different. IgG3 for the vaccine, this was actually above the upper limit of quantitation, and we chose not to run the assay again and do the dilutions, but it was off scale, so I'm just making the comment for that. But again, we see the same thing here for this particular host cell protein. It's different from the others, as well as this one. This one's lower. So we see, based upon the host cell protein, we're actually getting differences in the class switching that we're seeing. Now, interestingly, IgG4, which is considered to be somewhat of a regulatory antibody type, what we see is that this one, it's very high, it's statistically different from these, and number 7 and number 11.2, or is that 3? It's 11.2, are different from the others, but this one, 8, is also very, very low, and that's also the one that's highest in the other 3 too. And so what we saw is that there are differences in expression. So when you give different host cell proteins, you're going to get a different outcome. And not all of them are going to be immunogenic. Just because you give a host cell protein doesn't mean it's a problem. But we were able to show in the mice that they had different responses and that they were able to respond, which really surprised us at the time. And so in summary for that, for our host cell protein testing, we did show that they could activate and induce maturation of the immune cells in the mice. And small amounts, the 100 nanograms per injection, and we based that on how much we found we looked at different drug products that were commercial and how much host cell protein was in them, and we tried to make it an equivalent amount on a weight basis for the mouse, so that it was similar to what a person might see from actual drug product they received. But it could induce antibody class switching. Now again, we don't know that they were antigen specific, but it does show that it activated them. And it does suggest that maybe host cell proteins could act as an adjuvant or in some way induce anti-drug antibodies in context of the actual biological drug product that you're testing. And so with that, I'd like to thank lots of people in OPQ who helped us get those host cell proteins, and Daniela, Eric, and then people who have worked in my lab, and then Alexander and Shek, who gave me the information on the microphysiological systems. And with that, I'd like to say thank you.
Thank you, Kristina. That was a great overview as well about how to apply preclinical models to assess immunogenicity of some of the impurities in the products. The last talk that we have in this session is going to be given by Dr. Nico Gaudenzio. He is the research director at INSERM and a chief scientist officer at Genoskin. His expertise is in identifying molecular and cellular targets involved in allergic and non-allergic inflammation and developing novel analytical processes and systems to study preclinical models and clinical samples from patients. Today, he's going to present a new in vitro framework for assessing immunogenicity using comprehensive profiling of injectable human skin modules. This is a really exciting data that we have on human tissue, on how to use this type of tools to assess immunogenicity of genetic products and therapeutic proteins. Thank you, Nico.
All right. So first of all, I would like to thank Daniela and the organizer for the very kind invitation. So today I'm going to come with my Genoskin hat, basically, and to show you what we are developing in the company, and that might be useful actually for immunogenicity. Okay, if I can move forward. So this is my disclosure. And so Genoskin is a French-American company. We are based both in Toulouse, France, and out of Boston in Salem. We have been created in 2011, and we are performing, experiment for pharmaceutical partners and also non-therapeutic industry and for academics as well. We really focus on immunogenicity and analysis on how drugs actually would be a risk to trigger immune response, but at the site of injection. And this is what I'm going to show you today. Sorry. Okay, so the skin is really a complex organ, like all the human organs, basically. But what we are really interested at Genoskin and in my lab as well at INSAM is to study the human natural immune response which reside within the tissue. We know that blood circulating immune cells are not the cells that would be doing the job, basically, usually, but they need to infiltrate the tissue to have an to exert their activities. And the skin is a good example of that, as is the site of administration of many, many drugs, as you may know. In the skin, we can find, I'm going to show you, but almost 15 to 16 individual subpopulation of immune cells, which ranges from lymphoid cells to myeloid cells, which comes to different flavors. We have dendritic cells within the epidermal layers. We have many subtypes of dendritic cells within the dermis, macrophages, mast cells, which are also a very, very important component that have been often forgotten by immunologists and that can cause really a strong pseudoallergic reaction within the tissue. And there are many lymphoid cells, such as CD40 cells, innate lymphoid cells, regulatory T cells, CD8 T cells, NK cells, etc. So the idea is really to be able to study what's going on at the site of injection without extracting the cells from the tissue, because we know that it's really important for them to reside into their natural anatomical niche, because they are not functioning individually, but they do have a strong interactome. They form an ecosystem with the structural elements within the tissue, keratinocytes, stromal cells, and they all feed each other and talk to each other. So at Genoskin, what we do is that we have developed a network of surgeons and hospitals, both in Europe and in the US. And we are sourcing skin from tummy tucks, abdominoplasty, from former obese people that have stabilized for more than a year without comorbidities. And we take those surgical biopsies, which are normally discarded. And we have found a way to actually maintain the biopsy alive for a prolonged period of time. And I'm going to show you it can go up to 10 days, for example. So what we do is that from a large skin biopsy, we punch out explants, so what we call individual models. And they are full thickness biopsies with the hypodermis, the dermis, and the epidermal layers. Keeping the hypodermis is also super important because it's full of nutritive elements and it's also full of immune cells. And those biopsies, they are then encased within the matrix that is composed of human proteins and that maintain a physical, mechanical property of the tissue. And then on the top of that is glued a silicone ring to maintain the natural tension on the surface of the skin. And this is actually a critical component, maintaining the biophysical properties of the tissue. Because if you take an explant, and I'm sure some of you have already experienced that, and you put them directly in the culture medium, after 2-3 days, they're going to soften and they're going to become like a chewing gum, basically. And if you put any immunogenic substances, then Langerhans cells, dendritic cells will actually escape from the tissue and they will float within the medium. So here, what we found is that, and that is coming from many years of R&D, is that if you maintain those biophysical properties of the tissue, you can vaccinate the tissue, you can add immunogenic stimulation, the cell will not escape the tissue, actually. And then all of this is embedded within the culture medium that is chemically defined. And then we then treat and inject those modules I'm going to show you. And then we can extract many information using classical histology, cytokine dosage. We can do omics at the tissue level and at the single cell level. So we usually recommend to use the model up to 7 days. It can be more than that if you have a young donor. It can be a bit less if you have an older donor, basically. And you can test multiple routes of administration. We do a lot of sub-Q because most of the drugs are actually going sub-Q, but we can do as well intradermal or transdermal. And what we are doing on a business perspective on a daily basis is that we assess the risk of a molecule before going into phase 1 to be at risk to trigger injection site reactions in patients. So what I'm going to share with you now are biological validations, because if you want to use this kind of system, you need to know what are the limits of the system. And this is something that has been just accepted in the European Journal of Clinical Immunology and Allergy. And this is extracted from the article. So if you're interested in getting more and more detailed results, I encourage you to read it. So basically, sorry, and I didn't say too much. So basically, this is a histological section of the same of different modules generated from one individual at day 0, day 7, or day 10. And this is the epidermal layer, the dermis, the hypodermis, and what you can see is that we can maintain actually the 3 type of layers over the days. And if you notice, there is a thickening of the corneocytes on the top, because obviously we are not washing out the skin, we are not exfoliating as we do on a daily basis, I hope. And so you can see that the keratinocytes are actually proliferating. Then if you analyze more at the structural level using large field 2-photon microscopy, what you will see is that in yellow are the immune cells, in blue are the Claudin 1, important tight junctions of the epidermal layers, and in red are the blood vessels, and in green are the nerve projections. And what you can see here is that between day 0 and day 7, we don't see actually differences in the topography of the cells, and they are still there within the tissue. But it's not because we detect them within the tissue that it means that functionally they actually maintain over the days. So we then switch to single cell RNA-seq analysis. So we dissociate the tissue, we analyze the transcriptome of each individual cells of the tissue, structural and immune cells all together, and we analyze whether we're going to have transcriptomic evolution within the model over the 7 days of culture. So this is the typical picture of single cell RNA-seq analysis of a natural human skin. So you get 50% of the cells will be keratinocytes. That comes to also different subsets, such as undifferentiated, proliferating, differentiated keratinocytes. You will have fibroblasts, melanocytes, then lymphatics, endothelial cells, and myeloid and lymphoid cells. So what we did here is that we took from the same donor, we perform a single cell RNA-seq analysis at day 0, day 3, day 5, or day 10 of culture, and we compared the transcriptomic profile of all the different subsets that I just showed you. And here is the aggregated data sets. And what you can see already is that there is not the pink, which is day 10 on one side, and the gray, which is at day 0 on the other side, meaning that there is not a massive transcriptomic evolution that goes during the culture period. And here we can mathematically actually compare that. You can do a Pearson correlation by turning in each individual data set into a pseudo bulk, and then analyze how the transcriptome actually from day 0 will correlate with day 3, 5, and 10, etc. And what we could see is that we have more than 0.9 of Pearson correlation between day 0 and day 10, meaning that the transcriptomic profile overall of the tissue is actually maintained during the culture period. When you look at the proportion of the different cells, and also you can have variability depending on the dissociation of the tissue as well, but globally what we saw is that if you look at the red arrows here are pointing out toward the immune cells, up to day 10, we can actually maintain lymphoid and myeloid cells within the tissue. The only thing that we see is that after day 10, we start to see that a fibroblast might start actually to de-differentiate in the tissue. So if you want to use this model actually to study fibroblast differentiation, we would recommend to use them actually before day 7. So this is how it looks like. This is the model here, and this is an example of intradermal delivery or sub-Q delivery. So you can literally, if you do sub-Q, for example, you can literally take a forceps, pinch out the skin and then inject it like you would inject a patient. And this is how it looks like, the sub-Q. So this is a blue agent that will, it's contained, so we have a defined volume for which we know it does not leak out from the tissue. And then after 2 hours, 3 hours, it's going to start to biodiffuse in all the layers of the skin tissue. It's not then contained within the subcutaneous tissue. Here is, if it works, yeah. So here is a radiocontrast agent that has been injected. It's a 3D tomography. It's the sub-Q compartment, and here is the dermal compartment. So you can see that the liquid is really in the sub-Q space, and then it started actually to biodiffuse within the upper layers right after the injection. So then we wanted to really assess whether the immune cells were functional within the tissue. And then for that, we had a very simple approach. We inject within the tissue either saline or a vaccine as a well-characterized immunogenic stimulus, and we used the Moderna vaccine for COVID-19. So this is the experimental protocol. So we take the biopsy. From one individual donor, we can do almost 50 individual modules, meaning that we can do replicates in one donor. You can do longitudinal analysis. So over time, you can test different doses if you want to as well. You can use some of the modules to do proteomics, other to do transcriptomics, etc., flow cytometry, whatever is needed. And then we inject those modules with the vaccine, and then we analyze at 2 different levels. What's going on at the tissue level, that's if we take into consideration the entire skin ecosystem, or what's going on at the single cell level in immune cells. So here is an example of transcriptomic analysis at the tissue level by bulk RNA-seq analysis. So what we did is that we analyzed at 8 hours and 24 hours the number of genes that were differentially expressed after the injection of the vaccine as compared to saline. And what we could see is that we had 65 genes that were differentially expressed at 8 hours. However, if you wait 24 hours, so for the vaccine to well diffuse and be uptaken by the tissue, you will dose more than 300 genes that would be differentially expressed. And when you analyze those genes and infer biological pathway, you will find that these are genes involved in skin homeostasis, detection of a xenobiotic agent, so a foreign agents that penetrate within the tissue. And then we'll have also different consequences on cell adhesion, migration, of your modulation of blood vessels, neuroinflammatory processes. We know that vaccine is actually painful at the site of injection as well, and pro-inflammatory cytokine and immune cell activation. So this is just to show that even the injection of a molecule that is dedicated to target the immune system actually affects the global skin ecosystem, and that the skin is full of sensors. TLR, pattern recognizing receptors or any detection system are not only obviously restricted to immune cells, but the skin is full of those receptors, and this is what those data are actually showing as well. So when we switch then at the single cell level, so which immune cells are actually targeted by the vaccine at the site of injection? And again, this is the example with the vaccine, but it can be done with any type of immunological stimuli. When you isolate CD45 positive immune cells, this is the type of picture that you get in the human skin. So you can see that you have activated memory CD4 T cells, resident Tregs, mates, activated CD8 T cells, NK cells, mast cells, Langerhans cells that comes to 2 different flavors, a cluster of DC macrophages, and a cluster of migratory IPC, which are actually dendritic cells that are already activated. And what we did first is that we wonder actually which cell would incorporate the LNP particles in the vaccine. And for that, to analyze that, we did a very simple experiment, is that on the human genome database, we had a 25th chromosome that correspond to the spike sequence. So when we match our single cell data set, then we can detect actually transcript linked to the mRNA, spike mRNA, and to see in which individual single cell is actually expressed. And when we do that, we found that almost 60% of the vaccine would go into mast cells, while 40% of it will go into the dendritic cell macrophages clusters. And we found that very interesting because my lab at INSERM as well study mast cell, and they are associated with so many mediate allergic reaction at the site of injections, and the fact that most of LNP particles actually can target mast cells is kind of interesting in this topic. So then what we can do is that not only we can detect in which cells it has been incorporated, but we can also detect how the vaccine or the immunogenic substance has modified the transcriptomic profile of those cells, whether they are activated or not, etc. And what we could see is that at 8 hours, we have obviously dendritic cell macrophages that are the most affected by a vaccine injection, but also mast cell, CD8 T cells, and the cluster of Langerhans cells as well. While at 24 hours, the only immune subtypes that basically have a sustained activation and a sustained transcriptomic remodeling are actually the dendritic cells and the macrophages. So then when we look at in those dendritic cell and macrophages, how they are affected by the immunogenic stimulation, we could find that after the injection, you have activation of detection system, TLR-mediated signaling, obviously, NLRP3 inflammasome, clathrin-mediated endocytosis are activated, and we know that it's the major pathway for incorporating LNP particle into disease. Then we could also see, MHC class 1 cross-presentation activation, MHC class 2 antigen presentation as well, translational machinery and trafficking, obviously, endoplasmic reticulum Golgi, and activation of cell migration, antiviral responses, and basically there were modification of the global homeostasis of the dendritic cells. So we can really go into detail and see if we inject an immunogenic substance, which cell will actually are likely to be the most affected by those substances. Then we recently developed, and that has been published in Science Advances last year, a new multiplex imaging system that enabled to cartography the immune cells in 3 dimensions within the human skin, and that enabled you to map actually their activation status, their location, etc. And so we apply that to understand how the immunogenic substance would affect the immune cell at the site of injection. And this is how it looks like. We designed an antigen presentation panel full of co-stimulatory molecules, migratory molecules such as CCR7, that is required to migrate to lymph nodes. And what the software does is that after the 3D acquisition, it's going to create a matrix in which, depending on the combination of marker, it will classify your cells into Langerhans cells, dermal DC, Langerhans positive dermal DC, other cells, etc., and quantify the amount of activation markers that they have on the surface. And when we did that, we saw that the vaccine preferentially target the activation of Langerhans cells and Langerhans positive dendritic cells, but to a lesser extent, actually dermal DCs. And what was really interesting is that we can transform those data into kind of a Google map of the tissue where each individual immune cells will be mapped and with their activation status. And this is how we found that after vaccination, you have a cluster of myeloid cells that would actually aggregate around blood vessels within the tissue. Obviously, blood vessels are collapsed, so they cannot enter the vessel, but they would aggregate that and they would upregulate activation markers and migratory markers, such as here in green, CCR7. So to summarize, we think that with this system, we can analyze at the tissue level how a molecule can affect the global homeostasis of the tissue, understand what cytokines are produced. I didn't show you, we did also proteomics analysis, and what are the different pathways that are modulated, and then we can switch to the single cell level to understand for some molecules which cell are incorporated, the substances, how it affects the interactome between dendritic cells and T cells in the tissue, and globally at the protein level, the location of the cells. So that's pretty much it. Then we also conduct small clinical trials in which we can have up to, for example, 50 individuals that are analyzed, and then you get data, different doses, etc., in a cohort of donors. And with that, I would like to thank you with your attention.
Thank you, Nico. Those are very exciting ex vivo tools and analytical tools that I'm pretty sure that they're going to be the future of understanding immunogenicity at the local site of injection. Now I'd like to invite all the speakers to join us in the podium for a panelist discussion. We're going to have 30 minutes for question and answers. So. All right.
Are you sure?
Let's get started. I guess Mike has the microphone, so.
Sorry, I have it first. I'm going to ask Vibha a question, but basically I was really impressed with your presentation because you proved to me that host cell proteins are something that are of low risk. I mean, you had a product, GM-CSF, had E. coli, had antibodies against E. coli, and you didn't break tolerance. You take host cell proteins, put them in vitro, and you don't see any responses. You have a product that has that PLB, whatever it was, that was a contaminant that you could have responses against, and it didn't make any difference on the anti-product antibodies. That's 3. And then the last one is you had this really broad range of host cell protein contaminants, contaminants, let's say host cell protein content, from 100 ppm to 4,000 ppm. You go into clinical trial and you don't see any difference in safety, adverse reactions, or I don't think ADA, but you didn't do anti-HCP, but you did maybe anti-product. So you convinced me that host cell proteins are like really low risk. I wonder whether we even should be chasing, this moving target of something that seems obvious to me and to you, I think. I didn't see it on your summary, what you showed, but to me, it convinced me that host cell proteins are like limited risk. What's your comment?
You redid my talk again, but that's great. So I think you got all the good messages. So I just want to say one correction or one clarity. The 4,000 ppm material was tested only in vitro. We would never go with that kind of material in clinic. But what I was trying to show was that we worried about minor changes from lot to lot in host cell proteins? And is that something we need to stop, like purify or even terminate the entire lot and then start fresh? Because some companies are very worried about the guidance and they follow it exactly to the point. So I think when we did this analysis at Amgen, we were trying to show that if from phase 1 all the way to pivotal material, we are going to see these changes, are we really worried about the minor fluctuations? And is there a way for us to de-risk them? And instead of trying to go and develop anti-HCP assays in clinic and take more samples from patients and develop another assay, which we would then provide to the agency, which would show no relevance, we wanted to, in a way, show that these host cell proteins may not be increasing the risk. So I do agree that in our examples, we were able to show in vitro that they were not risky from an in vitro T cell proliferation assay, and also the in silico had identified these epitopes. So we did not want to test them in isolation. So if you notice our experimental design, when we looked at the drug product, we looked at it in association with these host cell proteins. If we had now done the assay with just host cell proteins alone, I think we would see cells proliferate, because then you are giving a cell an ability to take an antigenic sequence and make an immune response. So our intent was, how relevant can we be? So from that perspective, yes, when you're giving a host cell protein in minor amounts with your own biologic, which is predominantly going to be seen by the immune system, we are hoping that intrinsically, whatever that drug product does, you would see that, which is the profiles I shared with you. But from a host cell protein, you did not increase that risk. So I agree with that. But what I would like to say is that if we can do these kind of connections more and more as an industry and start collecting our clinical data and the lot pairing and start using that almost like making a machine learning tool, we may be able to get away from doing a lot or get concerned about these going forward. And subvisible particles and aggregates are a great example of that. We have spent almost 10 plus years trying to tell that subvisible particles are not a risk. And that was, again, Amgen did a lot of work with FDA in bringing all that new data, and now we know that they may not be problematic. So I would, again, reiterate that yes, host cell proteins at low amounts may not be risk when you're looking at a big, like, in a context of a protein or even a big formulation, but we have to de-risk it with some of these tools and add the data from clinical.
Can I have one more quick question after that? Oh, sorry.
Quick.
Okay, for Kristina. You're not off the hook. Okay, so you showed us a very complicated model of making a humanized mouse, which is difficult to do. And then you showed us data where you gave it peptides or other host cell proteins. And you didn't have a way to measure antigen specificity. You measured B cell numbers or plasma cell numbers. And to me, that's not really convincing that that's something to do with ADA or antigens. And then you went to show that if you sprayed in the nose, you got this inflammation that was an excipient, not really the product. You said it was an excipient. And I would make the case that everything you're seeing is an excipient.
Okay, so I agree with you, and as I mentioned during my talk, we did not do specific ADA responses. At this point, my lab is now doing that, so we can see neutralizing ADAs, but I have not done that with host cell proteins nor with generic peptides at this point. But I agree with you, without having something that's antigen specific, you don't know whether or not it is or not. What we were trying to show with those experiments was whether or not you could see anything at all. Would the model tell you, could the model tell you anything? It doesn't mean it's specific. Now, when we got the signal for the salmon calcitonin and we then followed up on it, that's when we took it apart and said, well, what if it's the excipient? What if it's this? What if it's that? And that's how we were able to figure out it's not the product. And so again, I would just look at it as being a tool to say, hey, we figured out what it is. It gave us a way to separate this to know what's causing the issue, rather than saying, hey, it's giving us a signal for everything, because we didn't see that. But I agree that we would need to have specific antigen responses. The other point that I would want to make, and this is a follow-on to Vibha's comment, just because you see activation in response to host cell proteins doesn't mean that you're going to have anti-drug antibodies to a product. And it doesn't mean that the host cell protein's a problem. I agree with that. But I do think it is important for us to get more data, as Vibha said, so that we know for sure exactly what we need, and then we can stop chasing it if it really is nothing.
Thank you.
So this is for our Vibha again, and this regards the PLBL2 response in CHO, and there's 80% homology of human PLBL2 to CHO. Now, PLBL2 is an intracellular protein, so even if those antibodies were to cross-react to human, they may not access the PLBL2 within the cell. But if there's trauma, infection, even apoptosis, is that an issue for these kinds of antibodies?
I think yes, and I think that's where all our questions, which are recent, which I shared with you for the cell and gene therapies, are coming, that we are going to express, if we have an engineered domain with any of these attributes, like CRISPR, or which is carrying over, we are expressing them intracellularly and then bringing it in the context of class 1. So I think, again, the same question, and if we are going with our CAR-Ts, for example, into lupus and multiple sclerosis, and there the reaction may be very different than what we have seen for a very oncology patient. So again, risk assessment needs to look at those components, and again, what we can do upfront, yes, great, but clinical data sometimes tells you a lot more. So we do change the risk. So one thing that we did want to do was specifications and adapting or changing the control ranges around the specification, and that only comes by including the new clinical experience. And we've made something which we called out as conservative to more flexible after we saw the phase 2 data, for example. I would not say phase 1 in biologics is good because it's single dose sometimes, but once you do phase 2 dosing with a higher dose and chronic dosing, and you don't see much happen, then the practice should be to go back to your CMC or process development and say, let's loosen these specifications because we're not seeing adverse events, and vice versa with diseases which are compromised, you have to look there too.
Just to follow on, I think what I've seen also here, Vibha, it's me. I've seen that in this same example, sometimes you have an excess of residual host cell protein in a product, and what happens is that indirectly it chimes in or it degrades the product per se, and then you end up having product variants that then increase the risk of immunogenicity for that particular product, because you may be exposing neoepitopes or epitopes that were not exposed in the original material. So I've seen in those instances where your risk assessment actually informs your quality control strategy and how to control for these product variants. And you really need to use these immunogenicity risk assessments to support comparability of the new material. So I think it's another layer of discussion. Is not the host cell protein per se that triggers the immune response and how this immune response modulates the response to the biologic, but it can indirectly have an impact by altering the quality of the product. Would you agree with that? Have you seen examples of that that can be shared with the audience? Because I have seen that.
I have not been directly involved with it. I have read about it, like what you're saying. So one thing that we do want to do is more and more relate whatever risks we think, like whether they're theoretical or some which have been identified, how do you understand what they do in clinic? And that's why I keep on going back to, like, is there a way for us to mine the clinical data in a more knowledgeable or informed way? And for that, we have to work closely with our CMC colleagues or process development colleagues, because if we think that those degrading fragments or even the HCPs are inducing those, what is the way to collect that information in vivo, right? And I remember at Merck, we were working on something called in vivo multi-attribute profiling. So it was almost doing maps like an assay, but in vivo, where you're collecting all those, what happens to drug product as soon as you dose, like whether sub-Q or IV. Can you detect the differences in the patterns? So again, it's disposition and how fast it gets disposed from sub-Q versus IV. And we were able to do that MAM profiling and get some knowledge from there. But again, how do you bring a specific attribute and allocate the adverse event to that specific, unless you have a high dose of it or high. So it's all about monitoring adverse event. There is a very good work from Alethea Bio from Europe. You may be aware of it, Tim Schuchman, who has done soluble HLA-associated peptides, which they look for like after dosing. And they've done it with cancer neoantigens. And now we are exploring it for looking at antigenic peptides. Can you detect them in vivo? And I think those are the things you need for correlating with maps or our ex vivo data, and then say, yes, we are seeing the same happen in vivo. But I do agree, and I've heard about it, what you're saying.
Sophie, you had a
I
comment.
had a question/comment to all these, all the thoughts that were just shared. As you say, it requires a lot of data to be able to do this, and I'm sure like several companies are collecting this data. Is there a way to put this together, or because I remember there are consortia, for cataloging those cell proteins from CHO cells, for instance, right? Is there a way to share the knowledge and be able to use ultimately AI or machine learning, etc.?
So I know all of us within our own organizations are working on it. When I was leaving Amgen was one of the first ones to make a tool for mining data from clinic and associating with lots. We are doing it now, so I feel that there are multiple ways to do this. Innovation and Quality Consortium, which works very closely again with FDA in terms of bringing the knowledge back to agency through didactic presentations, would be one way, and we could propose a consortium around it. There's a CMC regulatory leadership group within IQ who can do it. I'm part of a translational ADME leadership group now where we are talking about novel modalities. So definitely there are options to do this with all the industry partners being in a consortium and sharing.
Great. Danny?
I have a question about maps for Emilee. Hi, Emilee. Thank you for the very nice talk. I was looking at, well, I was not looking at the sequence, but one of my team was looking at the sequence that you showed that was the promiscuous epitope, and we recognized that epitope, and we were wondering if you had done some T-cell responses to that epitope.
So I feel like I've heard that question from you before, Annie. So yes, I think you are familiar with that particular sequence. The nice thing about our version of the T-cell proliferation assay is that you can use different T-cell markers. So you could certainly look to see if there are regulatory T-cell markers associated with that. We didn't go on to do that with that specific work to actually include those T-cell markers, though.
Right, so what I was thinking about when you said, do you want to pair this assay with another assay? You said MHC binding. I think T-cell assay or in silico, because the in silico would actually show you that that sequence is a T-regitope. So, but great presentation, thank you.
Thank you. Who has the mic? Oh, okay.
I have a question related to recombinant peptide. So, why clinical studies are required for recombinant peptides if we can show the sameness with the RLD, means as a generic company, and we characterize any impurity of the host cell peptides impurities. So, I'm not sure what is the logic behind doing the clinical studies, and if, as per the current regulation, we have filed ANDA using the synthetic peptide, and tomorrow we do post-approval changing the API source using recombinant. Can we do a supplement of that ANDA? So,
I will not venture into giving regulatory advice or on regulatory pathways. I think you should reach out to FDA because, as I said, I think we're always open to scientific justifications. So it's not, again, there's no regulatory mandate about what pathway specifically you may use for recombinant peptides. So it's all about you should contact FDA.
Okay, we understand right now there's a mandate. The guidelines clearly says if it's a recombinant API, you have to file 505(b)(2). So, because even if I submit the pre-ANDA meeting for the recombinant, they will say this is for 505(b)(2), go to NDA or OND. So,
yeah, well, I would encourage you to reach out.
Okay.
Yeah, sorry, I cannot really address your question.
Yeah. My other question is for alternative methods. So I'm not sure in those methods, we have not seen any advantages of the current in vitro assay methods we are using. What are the advantages? Is it with respect to economical, acceptability, or the time? So if somebody can guide us that. Right now we're using in vitro method for adoptive and innate. So these alternative methods which we discussed here, whether it's a human, mice, or this, the other one, where we talked about this in better methods which are under proposal. So what are the advantages we have?
So just so I understand your question, are you saying that the in vitro methods you're currently using you're not happy with and using the other methods?
No, I'm saying because we are discussing about the alternative methods, like for this humanized mice or this injectable human skin model. So I believe these are alternative methods to the current methods which we are using for adaptive and innate.
Yeah, I guess my comment on it would be to look at these from my perspective, and certainly I will allow the person who spoke on skin to give theirs. these are really orthogonal methods. So for example, if you get a signal from something and you're trying to determine, or you're trying to make an argument, hey, this isn't a problem, this really isn't a signal, these would be instances where potentially one of these methods might be useful depending upon what your product is. So what you had yesterday when we were having the group discussion, you had pointed out that, okay, you have a signal and there's really nothing wrong with the product. How do you prove it? Well, if the method that you're using shows a signal, then potentially having an orthogonal method, whatever it might be helpful to show that, no, there really isn't a signal, we can ignore this. And so I would not look at any of the alternative methods we're talking about here to replace in vitro. I would look at them more as adjunctive or informative to help show that there really isn't any risk, or to try to better understand the risk. So in the example I was answering earlier, we saw that signal when we gave the intranasal Thalmin calcitonin, and we immediately said, okay, well, which part of it, is it the actual API or is it something else? And when we took it apart, we said, oh, it's the excipient that's doing this, not the API. And so looking at these tools as basically a tool to dissecting if an issue comes up on one of the standard methods that you're doing right now is the way I would see employing them. Pass it over.
Oh, I totally agree that need to be seen as an additional detection system, not to replace the current in vitro testing. But for example, on our side, the skin is a full ecosystem full of pattern recognizing receptors. So you're not analyzing one immune cell, but you're analyzing like the entire tissue. So we think in terms of sensitivity, we can go a little bit further with that. Also, something important is that most of those molecules will be injected. And so with these, we can also mimic the injury of the injection at the site of injection. And I think it's very important as well. And when we talk about even traces of bacterial product, those skin are full of microbiome on the surface, which is the natural microbiome of each individuals. So I'm sure that even the injection, even if you have a super clean product, the injection through the human skin will cause a little bit of microbiome actually to go within the tissue and have traces level within the tissue at the site of injection. So we're still thinking about how to improve immunogenicity testing and take in consideration not only cells that would uptake the antigens, but really how the process of injection plus the product actually will favor immunogenicity.
Okay. So what I understand, these are not the alternative methods. This can be used for the reconfirmation of immunogenicity.
And to answer other questions, right? So that we cannot address with in vitro.
Sure.
Thank you.
Thanks.
Daniela?
So, Nicolas, actually...
Daniela, 1 minute. I think there was a response.
Oh, I'm sorry.
Yeah, this is Eric Pang from OGD. I'd just like to give a response to the regulatory question in terms of whether, 505(b)(2) is an automatic pathway for your recombinant peptides. I think the concern is that under the current synthetic peptide guidance, one of the footnotes says this is currently, we think that you need, if you have a recombinant peptide, we think you need additional clinical study to demonstrate sameness. Now, under this premise, you have to go to 505(b)(2). However, as all guidances, the recommendations, and if you do have justification or additional study or proposals, we'll welcome those, and you can present them, submit those, whether through a meeting request through us or even to the new drugs, to ask them, is clinical trial still needed if I provide these studies to justify that the risk is controlled? I think under that, if the response is no, or let's see what the data is, I think there could be room for discussion about having the recombinant peptide being submitted under the generic pathway. I hope that's helpful.
Thank you, Eric. Daniela?
So, I'm going to agree with Eric and just let the sponsors know that, what the guidance says, our recommendations, and you're welcome to bring any new assay. In fact, we love it when you do, and any new approach that might improve our assessment. But I wanted to go back to what Nicolas was saying. 'Cause, Nico, you mentioned in passing, you mentioned hypersensitivity, you mentioned in passing that your assay is good for looking at mast cells. So have you done any work into trying to assess whether your system can be used to maybe predict whether a product or impurities in a product could lead to hypersensitivity-like reactions?
Yeah, so we have been we have been injecting many drugs over the last 10 years for pharmaceutical clients. And in the R&D department as well, we investigated that. And notably, there is one drug named citrotide, that is given to women and subcutaneously. And it turns out that citrotide can be an agonist of one receptor on mast cells named MRGPRX2, which is a receptor for cationic molecules. And when women self-inject with citrotide, almost 10% of them get adverse event at the site of injection. And what we could see is that when we inject citrotide in a cohort of like 10, I would say, donors, almost half of them will develop a transcriptomic profile in the skin tissue that recapitulate features of type 2 immune response, detection of chemical substances, and blood vessel modulation, so all signs of edema, basically, and also neuroinflammatory processes as well associated with pain. And we know that when you trigger mast cell degranulation, you get edema formation and you get pain because they are strongly associated with neurons as well. So it's not a direct proof of hypersensitivity, but I think we might have a tool actually to detect, or at least to see which candidate would be at risk to trigger mast cell degranulation within the tissue. We actually have culture of primary human mast cell then ex vivo from CD34 progenitors, on which we can confirm direct activation of the substance of mast cell. And if it's not, then we can conclude that it's an indirect effect within the tissue through individual product on mast cell.
Yeah, we have a question.
So my question is to Vibha. Thank you for providing us a very good glimpse about what HCPs can be notorious and what can they possibly do when you inject HCPs alone, and you have shown some reactivities to these HCPs. So until now, we have been looking at HCPs from quantitative angle, 100 ppm being the max and so on. But we have never looked at it from a qualitative angle, where there are rogue HCPs and there are other HCPs that are not so rogue. Okay? So what should be the underlying kind of a way or approach should be to make sure that these HCPs are actually discerned and then looked at which are the rogue HCPs, and then limit those HCPs to a very low level, because these HCPs are either going to affect the stability of the product, can affect the formulations and so on. So it is time now to look at from a quantitative angle to a somewhat qualitative and quantitative angle for the HCPs, number one. Number two, and thank you, Eric, for providing a kind of a way or approach for looking at this, whether there is a safety to the recombinant products or not. And it is really a time to look at recombinant process and not take them through the clinical angle. Because this is a clear punishment for people following a recombinant pathway. And basically with GLPs and other kind of peptides which are in such a high demand, synthetic peptide chemistry is not able to reach that out. There is now a hope that recombinant can possibly make that difference. And the only problem here is the clinical data requirement, which is massive, and that's often a deterrent.
Again, it's not a requirement, it's what has been done historically, and we are here to discuss what we need to be doing, right? Not in 20 years, so we agree with you.
Yes, thank you, thank you. That is something that we all look forward to, the industry looks forward to, because that's where I think, and nowadays, there is so much of process improvements that has come around. The ppm levels, 100 ppm is too much. There are products with 1 ppm or non-detectable HCPs, okay? Which means the sensitivity of the methods is more limiting than than the HCPs alone. So it's time now to look at the overall risk behind the recombinant products versus the synthetic.
Thank you.
I encourage everyone to be on the lookout for the reopening of the public comments to our FRN.
Yeah.
So
Mohan?
this, sorry,
Mohan.
finish your thought. It's me? Okay.
Yes.
So this question is about the MAPS assays, and so possibly for Dr. Knowlton. So when you elute an epitope from the MHC, we have a great confidence that this peptide may be presented in vivo. Is the corollary true if you have a protein and peptides that are not eluted from the MHCs may not be presented in vivo? Do we have the same level of confidence to say that? I ask this because I want to know if we have standardized conditions to optimize the presentation so that all the possible epitopes are being presented, and also to see if data from vaccines where they have epitope reporters that are being presented, is that compared with the MAPS assays to have the confidence that whatever is not presented may not be presented in vivo? Thank you.
That's a great question. I mean, I think we know that some peptides just don't fly well in the mass spectrometer, so you're not always going to be able to pick some of those up. And also when you introduce that element of competition, there are a lot more sequences that are being presented. So again, you may not see some of those peptides that are presented at lower levels. But we've certainly worked with a lot of groups where we have detected those sequences that did go on to cause problems. So I'd suggest looking at some of those case studies.
I also have a follow-up question. Emilee, did you look at the sensitivity of your assays for the impurities? What's the level of impurity spiking or impurity?
Right. So that was kind of given as an example of what hypothetically we could do. So we haven't actually done the impurity spiking at this stage. So yeah, that's definitely something that groups will have to look into in more detail so we can understand the sensitivity of the assay.
Great, thank you. We have time for one last question.
Just maybe a question and a comment, and it's related to both the first question and the question of the gentleman here about there was a lot of numbers thrown about the quantities, quantification of HCPs, like 100 ppm. These numbers should be taken with a huge grain of salt because we don't really have any method to quantify host cell protein pool. It's like all these methods we have, they're semi-quantitative. So ELISAs, which are commonly used, polyclonal ELISA, they work well when you have the same HCP pool, the same sample from the same process step, and it can provide you relative quantification. So if you have like your ELISA shows you like say 100 ppm, it does not mean you have 100 ppm. Different ELISA would show you completely different number. It's just when you compare like 100 ppm and 200 ppm on the same sample, you know that you have maybe twice the amount of HCP. So setting, and actually no regulation or guidance have this 100 ppm number, no guidance set it. The USP mentions 1 to 100 ppm as sort of in the context of process capability. That's to the approximate level the process can reduce the HCPs, and the only other guidance I am aware of even mentioning this number is WHO guidance, and it also with a lot of caveats saying it's like roughly there. My question actually was for Vibha. You said you compared your 100 ppm level, or 40, I don't remember, in your product, and then you went 2 stage back in your manufacturing process and says, we have like 4,000 ppm. And my concern with this statement, it doesn't really mean that you have 40 times difference, because in the manufacturing process, HCPs clear differently, especially the problematic HCPs as PLBL2, they co-purify. So basically, potentially, you may have the same amount of PLBL2 in your final product as at your 4,000 ppm sample, because again, you don't know what HCP reduced at what manufacturing stage. So it should be comparing should be done very carefully. So that's my sort of comment about this whole business.
No, I agree, and everything is relative, but this is something that Amgen worked on. So there are 2 papers that are talking about an LC-MS, MS-based quantitation of HCPs. So they did actually went into the 24 very usual HCPs that were co-eluted with IgG2-based antibodies at that time. So we took 6 to 8, and the reason we narrowed down on those 24 were because every time we did a new antibody, they will always come up. And so LCMS was our main method. They were using ELISAs, but then they moved over to LCMS because they thought they could separate out these proteins and they could also quantitate. So there's a paper by Greg Flynn, who is a process development scientist, who has only talked about how he used LCMS to quantitate relatively, I would say. Yes, you're never going to be absolute. And then we did use that method to look at a FVIP column purified material versus the final formulated. And there were distinct differences in the overall ppms, yes, but we were looking at PLBL2 for sure, and there was a specific change in the amount that was purified for the final versus that FVIP, where we found more. And I think no method is perfect, and I know we should stay, this cannot be a black and white kind of a answer, but it was relatively, we could use those as controls in our assay to look for thresholds. So that was the intent.
Okay, thank you.
Thank you. And now I would like you to join me to thank all the speakers for such wonderful presentations. And I would like to thank you all for your participation in this session. And now we will adjourn for an hour so we can have lunch, replenish energy to hear about oligonucleotide therapeutics. Thank you.